Click for IB Biology. General principles of respiration, taking a look at chemiosmotic theory, process which occurs on the Cristae membranes and involves a knowledge of the electron transfer chain. A PDF of this presentation is available down here in the description, which you can annotate with your own notes, and there's a quiz as well to check your understandings at the end. Recall that from glycolysis, the link reaction, and from the Krebs reaction in previous videos, we've seen that the coenzyme NAD has been reduced. It has absorbed energy, usually by forming bonds to the hydrogens, and so we try to remember that the energy is in this structure here, and what we're looking at is how does that become transferred to form ATP. In 1978, Peter Mitchell was awarded the Nobel Prize for chemiosmotic theory and the electron transfer chain principles, in which the energy in reduced NAD, that energy is transferred to ADP to form ATP. And of course, in that process, the NAD is able to return back to glycolysis, the link reaction, and the Krebs cycle in order to be further reduced. How this transfer of energy takes place, so there's the oxidation of the reduced NAD is coupled to the reduction of the ADP. How this transfer occurs is to do with the structure found on the Cristae membrane. So the Cristae membrane involves these folds on the inner membrane, and students will recognize that with folding comes an increase in the surface area. And this is important so the Cristae can form these electron transfer systems. The incoming reduced coenzyme NAD dissociates its hydrogens, releases its hydrogens and electrons. The electrons are used to power 3 membrane pumps, and these pumps are responsible for pumping hydrogen ions into the space between the folded Cristae membrane and the outer membrane of the mitochondria. So this restricted space becomes important. The electrons power the first pump, pumping the protons into here. The electrons then pass to the second protein pump with more protons taken from the matrix into here. The electrons then pass to the third pump. Once more, the protons are pumped from here into here. And so by the end of this process, we end up with a high concentration of protons, hydrogen ions, in the Cristae space. So we have a high concentration. Mitchell then went on to propose that the protons would then diffuse through this structure here, which is called ATP synthase. So this is ASE is an enzyme. So the protons in some way were linked to driving the enzyme ATP synthase and generating and turning ADP with phosphates into ATP. The details of this mechanism and how this occurs is included in what is called rotational catalysis, and that's the subject of our next video. An important aspect of Mitchell's chemiosmotic theory occurs here. The electrons coming from the third and final protein need to be absorbed, and this is achieved through the combination of using hydrogen ions and oxygen and mopping up those electrons, and this results in the formation of water. This water is known as metabolic water. In this way, the transfer of the electrons in the electron transfer chain can continue as long as we have sufficient oxygen. And this is a link to another video in which we consider anaerobic respiration. It does also, however, draw attention to the function of the oxygen, which we most importantly continue to take in and deliver to our cells, that its function ultimately is to absorb electrons from the electron transfer chain. The links being shown next are to the mechanism of the ATP synthase, a subscription button should you care to subscribe, and also to the playlist where you can find out more details of the other videos in the principles of respiration.